All right, we're going to be moving on to chapter 3. And this is the first half of chapter 3, and it's going to focus on chemical reactions and enzymes. So the first thing we need to talk a little bit about is energy. And this chapter is going to be talking about energy a lot because in the second half we're going to be talking about cellular respiration, how we get energy to make ATP. Energy is the capacity to do work, and we kind of have two types, potential energy, which is stored energy or the energy of position, and kinetic energy, which is the energy of motion, and they can be converted sort of back and forth from one class to the other. So potential energy, if you think about like pulling a bow back and holding that the bowstring like that, that's potential energy, right? This is position, and then when you let it go and it twings forward, then it's going to be kinetic energy or the energy of motion. So there's a couple places where we have potential energy in our body. And one of them, and we'll be talking about this a lot, is that there's potential energy across the plasma membrane. And usually there's a concentration gradient. And this concentration gradient just means that there is a different concentration on either side of the barrier, the plasma membrane. So let's say we have a plasma membrane and on one side we have a whole lot of sodium and the other side we don't have very much sodium. We'll talk a lot about this in chapter 4, but the sodium is going to want to go from this really crowded place with lots of other sodium. It's going to want to go to the other side of the membrane where there's not as much sodium. And so that is, so if we opened a door in that, or a channel in that plasma membrane, your sodium would all rush through, and that's kind of a potential energy. We also have potential energy in our electron shells, and so electrons move from the higher to lower energy shell. So our higher energy shells are out here further away from the nucleus, and they want to get closer to the nucleus. So electrons in high energy shells that are further away have potential energy, and then electrons falling from those high shells have kinetic energy, and they're moving towards the nucleus. And we can kind of see like the electron is negative, right? And our protons in the nucleus are positive, so they're going to kind of want to be close to there. And here is a review of our plasma membrane energy, right? Where we have, here's our plasma membrane, and here's our high concentration of sodium, and they all have high potential energy because they're all, shoved together in this one little spot, and they don't want to really be so close to all the other sodium. So if this door opens, they want to go through. And so when that happens, the sodium ions that move to the area of low concentration have kinetic energy. So the stored energy here, the energy of position, is going to be the potential energy, and then as they move, that's going to be kinetic energy. Chemical energy is another form of potential energy, and this is a really important one. We have a lot of this in our body. We use a lot of this. Energy is stored in the molecule's chemical bonds, and it's released when the bonds are broken. And it's a little bit of a simplified view, but that's basically what it is. It can be used for movement, the synthesis of other molecules, and establishing concentration gradients. So we just said the concentration gradient was the difference between the concentration of a substance in two different areas, like on either side of this plasma membrane. And so this chemical energy is present in all chemical bonds, but molecules that are pretty important that we use to store energy as are going to be triglycerides. Remember, triglycerides were a storage form of what? Which biological macromolecule? A storage form of lipids. Glucose. And how is glucose stored? Glucose can be stored in the body as glycogen, usually in the liver and the skeletal muscle. And then ATP, right? ATP, we said, is that modified nucleotide that's the energy currency of the cell. And it has those really high energy phosphate bonds at the end that when they're broken, they release a lot of energy. So all of these are going to be important in our energy storage. So now we have a lot of kinetic energy forms in our body too, and I'm going to go over these pretty quickly. They're a little bit more self-explanatory, I think, although this first one here is pretty important. So we do have electrical energy moving through our body all the time, and it's basically electrical energy is the movement of charged particles, so the movement of ions especially. So the movement of ions across the plasma membrane of a neuron is a type of electrical energy, and this electrical impulse is going to be sort of the way that our nerves are going to be communicating to our brain. So our brain is really good at interpreting electrical energy, but not really the other types as well. Mechanical energy is exhibited by objects in motion due to applied force, like our muscles are contracting for walking. Sound energy, you have molecules that are compressed, and it's caused by a vibrating object. So we have sound waves that are going to come into the ear, and they're going to cause our eardrum to vibrate. Radiant energy, which is the energy of electromagnetic waves. So a good example of that is visible light striking the retina, right? And our retina is going to be able to interpret that. And a lot of these different types of energy have to do with our senses. Like, so this is our sense of hearing, our sense of vision, and we kind of need to have special receptors to understand these, or to accept these types of energy and transfer them to the electrical energy that the brain uses. And then heat. Heat is the energy of electromagnetic waves. Also, heat is usually not available to do work, and it's measured as the temperature of an object. So in chapter 1, we talked a little bit about homeostasis and how we use our muscles to generate heat in order to maintain body temperature. And we can see this is a little bit of information about the electromagnetic spectrum. So we have some visible light that the retina can perceive here. And then we've got this area, UV light and X-rays and gamma rays. These are going to be sort of those high energy rays, and they can cause mutations in our DNA. And radio waves are lower energy out there. We won't talk too much about the electromagnetic spectrum, but so now I want to briefly mention, this is brief, the laws of thermodynamics. So thermodynamics is the study of energy transformations, right? And we said that energy can be transformed from one state to another, but it can neither be created or destroyed. So it can only change in form. That's the first law of thermodynamics. So energy can neither be created nor destroyed. It can only change in form. But the second law of thermodynamics is going to state that when energy is transformed, right, so it's when it's changed in form, some of the energy is lost as heat. And so that heat is going to, usually we can't use the heat necessarily to do work, but in our bodies it's pretty important because we want to stay at a certain temperature. But when this happens, that energy that's lost as heat, we can't use that to do work. And so the usable energy is kind of decreased. And so as chemical energy converts to mechanical energy, heat is produced. And our mitochondria are going to help, like as they're working through all of the, what we'll talk about in the second part of this, and our aerobic cellular respiration, the mitochondria are actually going to be, changing energy around, and they're going to produce heat. And that's going to actually heat up the mitochondria, and that's going to happen a lot in things like skeletal muscle, and that's going to help heat up the cell. So metabolism is going to be the term. We learned about this in the first chapter. We said this is one of the things that all organisms have. They have metabolism, and metabolism is the term for all the biochemical reactions in living organisms. So it's the sum total of all the reactions. And these chemical reactions that we're talking about are going to occur when chemical bonds in existing molecular structures are broken and then new bonds are formed. And so usually we have the old bonds, and then we have to break those old bonds to form the new bonds. And this can be expressed as a chemical equation. So we'll say we have the reactants, which are the things that we start with, and the products, which are the things that we end with. So reactants are usually written on the left side of the equation. This is what we start with. So in this case, we've got the reactants here, A and B, and then the products are going to be what's formed by the reaction. They're going to be written on the right side of the equation. And so in this example, C is the product. And the arrow indicates the arrow of direction, so the reaction direction. So in this case, we have A and B are combined, and they form C, and it goes in one direction. In a balanced equation, the number of elements are equal on both sides of the reaction. We might have 2 hydrogens and an oxygen combining to form H2O. So we would have the same number of atoms and elements on each side. We can classify chemical reactions based on 3 different criteria. So we can look at the change in chemical structure, we can look at the change in chemical energy, and we can see if the reaction is reversible or not. So we'll first start looking at chemical structure. And we've got 3 different types. We have decomposition reactions, synthesis reactions, and exchange reactions. And we've talked a little bit about these already when we talked about our biological macromolecules and how we take those monomers and make them into polymers or break the polymers down into monomers. So hopefully this will be a little bit of a review. So this first part is a decomposition reaction. So whenever we talk about a decomposition reaction, we're talking about a reaction that starts with a bigger molecule and makes 2 smaller, or not necessarily 2, but makes more smaller molecules. So in this example, we have A, B, so 1 big molecule, and then it's going to break down into A plus B. So we're breaking it down. And so all decomposition reactions in the body together are referred to as catabolism or catabolic reactions. And so remember we said that cats like to break things down, so remember catabolism is the breakdown reaction. We talked about hydrolysis reactions, and we said the hydrolysis reactions were important in breaking down polymers into monomers. And so in a hydrolysis reaction, we're going to be adding water, right, to break down that bond right here. So this is an example here where we have glucose and fructose bound together into a disaccharide called sucrose. So we're adding water in this hydrolysis reaction, and we're going to be, so we're taking that oxygen here and we're adding, breaking that bond over here, and we're adding hydrogen on one side and we're adding hydroxide on the other side. And so now we have 2 separate monosaccharides, glucose and fructose. And so this is a good example of a hydrolysis reaction that we talked about in chapter 2. Now the opposite of this is going to be if we start with 2 smaller reactions here, or 2 smaller structures, and we combine them to form a larger structure. So A plus B equals AB. And so a good example of this is a dehydration synthesis reaction, right? It makes sense that a dehydration synthesis reaction is a type of synthesis reaction. And so the example that your book uses is once again from chapter 2, where we said amino acids are going to be formed by a dehydration synthesis reaction. They're going to be formed into a dipeptide here, and we've got a peptide bond formed in here. So a dehydration synthesis reaction where we are going to be, if it would be the same as this, we were going to be taking a hydrogen off of one side and a hydroxide off the other side, and then joining them together at that oxygen. It's another good example of a dehydration synthesis. So we call it dehydration because we're removing water. And then those are a little bit simpler to look at, but then we also have another type called exchange reactions. And this is probably the most common in the human body. And this is where we have groups that are exchanged between 2 chemical structures. And so it's got elements of both decomposition and synthesis reactions. And so we have AB plus C equals A plus BC. So in this case, we're moving the B from that first structure to the second. And an example of this would be a fast way to regenerate ATP in muscle tissue. And when we get to chapter 10, we're going to revisit this reaction. But so this reaction here, we have a creatine phosphate, right? And it's got this little phosphate group on the end. And then we have an ADP, adenine diphosphate, right? Adenine diphosphate is not as useful as ATP though. So what we really want to do is get that ATP back, right? Because remember, ATP is that high energy molecule, the energy currency of the cell. So we're going to have an enzyme here that's going to help, and it's going to take this phosphate group off the creatine phosphate and move it to the ADP, and now we have a creatine and an ATP. So we've moved that phosphate group over, and that's going to be an exchange reaction. So we'll come back and revisit this one again, this particular reaction again in chapter 10. And so we have lots of different types of these. This is probably the most common, where we're just moving things around. And so a specific type of an exchange reaction is called an oxidation reduction reaction, or you can call it redox reaction, right? So that's oxidation and reduction reaction. And so this is a specific type of exchange reaction where electrons are moved from one chemical structure to the other, and then they may be moved alone or with the hydrogen ion. So a lot of times you'll see that hydrogen ion move with them too. And so these reactions always have to occur together, right? So we always have to have an oxidation and a reduction because we're taking the electrons from somewhere and moving them to somewhere else. They're not just kind of disappearing, and we're not just getting them from nowhere, from thin air. So the structure that loses an electron is called, we say it's oxidized during oxidation. The structure that gains an electron is reduced during reduction. And you can remember this by the acronym LEO says GER. So Leo, L-E-O, Leo's a lion. So losing an electron is oxidation. Says Ger, G-E-R. Gaining an electron is reduction. So Leo says Ger to remember which one is which. So losing an electron is oxidation. Gaining an electron is reduction. Leo says Ger. And so this is important because we're moving these electrons. And a very big example of this is the nicotinamide adenine dinucleotide, or NAD plus over here. We're going to talk about this a lot in the next lecture when we talk about aerobic cellular respiration. And so NAD has different forms. It has an oxidized form and a reduced form. And so what happens is that this oxidized form right here can then accept electrons from other energy-rich molecules. And then once they accept the electrons, it becomes itself an energy-rich, so that it becomes reduced, and then that reduced form is energy-rich. So it's kind of holding on to that energy. So as electrons are moved, just like we kind of talked about that electrons have potential energy and the position of electrons is important, and as we're moving these electrons, you can think of it, it's like we're moving energy. And so at first we have glucose, which is an energy-rich molecule. Remember we said it has a lot of energy stored in its chemical bonds, and so we're going to oxidize that, right? So LEO, Leo, losing electrons is oxidation. And so it's going to lose its electrons, and it gives up 2 hydrogen atoms also, right? Because we said sometimes those hydrogens are moved with the electrons too. And then NAD here, which has already been oxidized, is going to accept these hydrogen ions, accept a hydrogen ion and 2 electrons. And so it becomes reduced. And as it accepts the electrons and becomes reduced, it accepts their energy. And then we have this energy-rich molecule. And then we also have a hydrogen ion that's formed. So that glucose has that extra hydrogen ion up here. So here, NAD is going to be the oxidized form, and it is able to accept electrons, but it's not energy rich, whereas the reduced form, NADH, right here, has accepted those energy rich electrons, and it has now become reduced, and it is an energy rich molecule. And we'll be talking a lot about NAD and NADH in the next lecture. So remember them, we'll come back to them. So we can also classify chemical reactions by the changes in chemical energy. So there's 2 different classifications here. We can call them exergonic or endergonic reactions. So exergonic reactions have reactants with more energies within their chemical bonds than the products do. And so that means the ones on that we start with had more potential energy, and the ones that we end with have less. And so that means energy is released. So most of these are going to be like decomposition reactions. And we can see right here, we've got an exergonic reaction. So we've got the reactants like glucose and oxygen. They have a lot of energy stored in their bonds. And then the products of cellular respiration are carbon dioxide and water. They don't have as much energy stored in their bonds, so the energy has been released. That's an exergonic reaction. Whereas the opposite of that is going to be this endergonic reaction. This endergonic reaction, we have the reactants have less potential energy within their bonds, and the products have more potential energy within their bonds. So the energy had to come from somewhere, so we have to supply that energy in order for this, usually in the form of ATP, in order for this reaction to go. And usually these are going to be synthesis reactions. And so I want to briefly talk a little bit about ATP cycling. Remember we said that ATP is the energy currency of the cell. And the cells are going to continuously form and break down ATP. It doesn't last very long. When energy is released in exergonic reactions, we said it doesn't just go away when we release the energy. We usually use that energy to form ATP. And ATP is going to then be able to like hold on to that energy, and we can, it makes it into a form that's usable by most of the things in the cell. And so fuel molecules from food are oxidized, and the energy in their bonds is transferred to ADP and a free phosphate to form ATP. And so that energy that's been released from endergonic reactions is used to synthesize ATP. And that ATP then we can kind of like move around and the cell can use it very well. ATP is then oxidized to aid in the endergonic reactions, right? We said endergonic reactions need energy to go, and usually ATP is what is going to be used to fuel that reaction. So the energy released from the hydrolysis of ATP, from taking the phosphate off the end of that ATP, provides energy in order to cause those endergonic reactions to be able to go. Now, our body is, like, ATP is a good storage molecule for energy, and it's really important that we have ATP and that the cell can use it really easily, but only a few seconds worth of ATP is present at any time because we're constantly using it and constantly making it. So we have to continuously form ATP to provide energy. And so here, this is our ATP. We have our adenine, our ribose sugar, and we have our 3 phosphate groups here. And this last one here is that high energy bond. When we split ATP, which is an exergonic reaction, energy is released, and that energy can then be used to fuel other endergonic reactions. And then now we have ADP, it has 2 phosphates on it, plus an inorganic phosphate over here. And when we have an exergonic reaction, remember we said exergonic reactions release energy, so that energy that's released from exergonic reactions can be used to form ATP. So we can take this ADP and this phosphate and bind them together, and that itself is an endergonic reaction. So we use the energy supplied from the exergonic reactions to form other endergonic reactions. Now the last classification of chemical reactions that we have is what deciding if the reaction is irreversible or reversible. And so you can look at the arrows in our chemical equation to try to figure this out. So an irreversible reaction is just going to go in one direction. So we have our reactants and they become our product. And then it can't go backward, right? So we have a net loss of reactants and a gain of products. And so it's kind of like if you are baking a cake, right? So you are taking all the ingredients and you're mixing them together, and then you're making the cake. At the end, you have less ingredients and more cake, and you can't really go back. You can't like say, oh, but I need more sugar. I want to take the sugar back out of the cake. You can't really take it back out that way. So it's irreversible. Once you've baked it, the ingredients are gone, but you have the cake. Whereas a reversible reaction is going to be sort of different. It doesn't only go in one direction. It can go in both directions. And so it's usually indicated by these double arrows here. And so we don't have any net change in the concentration of either reactants or products. They're going to be at equilibrium because we can have these two. Think of like Legos when you think of a reversible reaction, where you can take all of your individual Legos and put them together and build like a Lego castle. But once you're done with that, you don't, like, if you need more Legos, you can always break them back down and get the individual Legos back, right? So you can put them together and you break them apart. You put them together and you break them apart. And so, if you want to build more stuff, then, you just put them together and build more stuff. If you need to use more individual Legos, you can break it back down. And so one important thing to know and think about reversible reactions is that these are going to always exist at an equilibrium. And so we usually have about the same amount of A plus B as we do AB. And so think about, like, if you have an increase in reactants, so if we have a lot more of these individual A's and B's, we've got a whole pile of new Legos that, we haven't built yet, then it's going to kind of cause you to be like, oh, I really want to build more stuff, and you're going to make more Lego models, right? And so an increase in reactants is going to drive the equation to the right. So as you add more reactants, you're going to build more models. But then a decrease in products is right here, or a decrease in products will also drive the equation to the right. So if for some reason you don't have very many models that are built at all, you might want to build more models. Like, you have to, display more of them. So if you have more products or more reactants or less products, you're going to build more. If you have less reactants or more products, you're going to break down more. And so, like, if you have very few individual Lego blocks left, but you have a whole bunch of models that are built, maybe you don't have enough room for the models or something, and so you're going to break those back down again. And so you're going to kind of have a similar, like, pile of the A and B together, a similarly sized pile of reactants and a similarly sized pile of products, and they kind of need to stay in equilibrium. This concept is going to come back. We're going to talk a little bit about it again in 169. So just remember that these reversible reactions are going to remain in equilibrium. So an increase in reactants or a decrease in products is going to drive the equation over to the right, whereas a decrease in reactants or an increase in products is going to drive the equation over to the left. There is a good table in your book that goes over all the different types of, all the different classifications of these different reactions. So you can look at that table and that, use that to organize your thoughts. So next I want to talk a little bit about reaction rates. So reactions can either be fast or slow, right? So a reaction rate measures how quickly a chemical reaction takes place. And in order to figure out how quickly it happens, we need to figure out what the activation energy is. The activation energy is going to be the energy that's required to break those existing chemical bonds. So we said before this whole reaction happens, we have to break the bonds that are already there in order to make new bonds. And so we always need to add a little bit of energy at the beginning to break those bonds. And so this activation energy, it's a big E with a little a down there, is a primary factor to determine the reaction rate. So in order to start this reaction, we have to add some energy, and it sometimes takes time to do that. We have to overcome the activation energy. And so this picture right here is going to show us, this is like the activation energy here. And then eventually, so even if this is going to be an exergonic reaction where we're eventually going to release energy, we need to add a little bit at the beginning, this activation energy. And so this is that initial hump where we have to save up some energy. It's going to take a little bit of time before we can get enough energy in order to let this reaction proceed. And so in order to overcome the reaction energy, we need to overcome the activation energy, we need to provide enough energy in order for this to happen. And the faster we can provide the energy, the faster the reaction will happen. And in a lab, what we can do is we can increase the temperature. And by doing that, that provides energy to break the bonds. But significantly increasing the temperature in your body is not really that helpful because it's going to possibly denature those proteins. Remember, kind of melt the proteins and it changes their shape, and that is going to be not good for your body, right? You don't really need to have a really high body temperature all the time. And so instead of, increasing the temperature in our body, we use something called a protein catalyst, or an enzyme is used instead. And enzymes are going to be what we'll talk about for the rest of this lecture. So enzymes are catalysts that accelerate normal physiologic activities. So they don't cause things to happen that wouldn't already happen. But if those reactions would have happened, but maybe it would take a long time for them to happen, this enzyme makes them happen more quickly. And how it does this is by decreasing the activation energy of the reaction. And we call a reaction that's uncatalyzed a reaction that has no enzyme present, and then the catalyzed reaction has an enzyme present. And so this is going to increase the rate of the reaction and increase the rate of the products being formation. So in this case here, our example is an exergonic reaction, right? So we said we have sucrose and we're going to break it down into glucose and fructose. So this is that same example we used earlier. And so here is the normal activation enzyme that's uncatalyzed without the enzyme. We would have to save up, a lot of energy before we can actually have this reaction occur. But what the enzyme does is the enzyme is going to kind of group these. It's going to bind to the to sucrose here and kind of give it a hug, and it's going to change the shape of this area here, and it's going to make it so that it takes less energy to break those bonds. And so it decreases the activation energy. And so by decreasing the activation energy, this can happen faster. And I like to think of, let's say you're saving up for something. Like you want to buy, something from the store, and it's $100. And, let's say you're like a kid and you're trying to buy a bike that's $100, but you only get allowance. You get $5 a week or something. So it's going to take a long time, right? It's going to take 20 weeks to get your bike because, it takes a long time to save up that money. But what if the bike goes on sale right? And if the bike goes on sale and it's, oh, it's 50, because this looks like it's about 50%, let's say it's 50% off, well then if it's only $50 instead of $100, then it's only going to take 10 weeks instead of 20. And so it's going to happen faster, right? Because you don't need to save up as much energy, and so you can definitely save that up more quickly. And so by decreasing the activation energy, the enzyme is going to increase the reaction rate. So it makes the reaction happen faster. All right, so what exactly is an enzyme? An enzyme is a protein. So remember, we looked at biological macromolecules, and we talked a little bit about proteins and protein structures. And most of these enzymes are globular proteins. And a globular protein is a big sort of, it's not a straight, like, thin protein. It is a big sort of globby protein. So most of them range from about 60 amino acids to 2,500 amino acids. So they are pretty. There can be very small ones and very big ones, and they have the shape is very important. Like I said before, for proteins, the shape is really important to how they function. And so this has a unique three-dimensional shape, sort of inside the protein, called the active site, and this is where our substrate is going to bind. And so here, this is the enzyme. It's big and globby, and it has this opening, this hole called the active site, and it's the exact size and shape of the substrate. And so the substrate will fit right in there, and it kind of gives it a hug. And this means that the active site can only bind to this one substrate, right? Because the shape is different. So it only binds to the one type of substrate, and therefore each enzyme is only going to catalyze one specific reaction. And so it's going to, give this enzyme a hug, decrease the activation energy, and, cause the reaction to happen more quickly. So some of these enzymes are going to be within cells. So like DNA polymerase is within the cell, right? It helps form new DNA. Some of them become embedded in the plasma membrane of a cell. And so an example would be the brush border enzymes in the small intestine. So lactase is in the walls of the small intestine, and it helps digest lactose, as we drink it from milk. And some of them are secreted from the cell. So a good example of an enzyme is pancreatic amylase. It's released from the pancreas, and it helps participate in starch digestion. So enzymes are, how exactly do enzymes work? So how do enzymes catalyze things? And so enzymes, remember we said, they have this active site. And this active site is very specific, and it only binds a specific type of substrate. So that substrate, the one that fits, is going to go right there, and it fits like a lock and a key. It's going to fit into this active site, and it forms what's called an enzyme substrate complex. The enzyme then changes shape slightly, and this is going to stress the chemical bonds, and so it decreases the activation energy. It makes it easier to break those old bonds, and it lets the new bonds be formed more quickly. Then the products are released, and the enzyme is not harmed in the process. So it can catalyze hundreds of reactions, only the one type of reaction, but it can do it 100 different times, like many different times. And so here we have lactase right here. We said this is going to be embedded in the small intestinal wall, the microvilli of the small intestine. So it's going to fit well with this lactose. And so it's going to give it a hug like this, and it's going to stress those original bonds and kind of help break those original bonds, and then decreases that activation energy so that we can have the glucose and galactase are going to then be free. And now, as we can see, lactase is still it's still healthy, and it can then go back and catalyze another reaction. So it is not destroyed in the process. It also can be used in synthesis reactions. So here, like in the liver, when we're storing glucose as glycogen, we have the enzyme glycogen synthetase. And it's going to bind to two separate glucose molecules. And then remember that we have to take a hydrogen ion off one and hydroxide off the other in this dehydration synthesis reaction first, like break those initial bonds before we make that new bond at the oxygen. So it's going to change the shape a little bit, decrease the activation energy, help those initial bonds be broken, and then make it easier to form the bond between the glucose molecules. And then glycogen is released, and the enzyme is free to participate in another reaction. So it is important that these enzymes are regulated, that we regulate the enzymes and how they work. And one of the things that we use to regulate the enzymes or activate them is a cofactor. And cofactors and coenzymes. Cofactors are molecules or helper ions that are required to activate the enzyme or to ensure that the reaction occurs. So these are really important, and we have lots of different enzymes that require lots of different cofactors or coenzymes. So if we have, these are not proteins usually, but they can be either organic or inorganic in structure. So an inorganic cofactor is like a mineral, like a zinc ion or something like that. So these minerals are going to attach to the enzyme to help it work. And so each enzyme, is going to need different types of these cofactors. So an example is that a zinc ion is required for carbonic anhydrase to function. And this is an important enzyme that we'll talk about in 169. So we need zinc in our diet in order for this enzyme to function. And that enzyme is important for making, important reactions occur. Now, if our cofactor is organic, so if it has a carbon and a hydrogen, we call it a coenzyme instead. And so these coenzymes, a lot of them are vitamins. So like B vitamins are really common and very important coenzymes. Sometimes they're modified nucleotides that can serve as coenzymes. And so these coenzymes, like NAD and things like that, are going to be really important in, they need to be present in order for the enzyme to work. And so in order for all of those in order for all the reactions to happen as quickly as they need. So these are very important, and we need each different enzyme is going to require different cofactors and coenzymes in order for the reactions to occur, in order for them to be active. So once the enzyme is active, right, and we have the enzyme and we have substrate, we know that what does the enzyme do? It increases the reaction rate, so it makes the reaction happen faster. But if we only had one enzyme and we had a whole bunch of reactions that needed to happen, the reaction still wouldn't happen that fast. And so sometimes we need more than one enzyme in order for these to happen. So as we look at the rate of the chemical reaction, if we just added one enzyme, it's not going to increase the rate significantly. But if we added, 10 enzymes or 20 enzymes, it would increase the rate a lot faster. So the rate of the chemical reaction is going to be accelerated by a couple of things. So we can make the chemical reaction faster by increasing the enzyme concentration. So we need to add more enzymes. And so if we add more enzymes, that's going to make the reaction happen faster because we have more enzymes that are ready to, catalyze that reaction. And so here we can see that the rate of the reaction is going to, this next part, sorry. It makes sense that if we add more enzymes, the rate of the reaction increases. But no, we can also increase the rate of the reaction by adding more substrate. And the substrate is the stuff that the enzyme uses, right? Remember that enzyme substrate complex. So if we had, say, 100 enzymes, but we only had, 10 substrates, adding more enzyme is not going to help because we don't have enough, substrate in order, for all some of the enzymes aren't busy already. And so if we have, a whole bunch of substrate and we have only a couple of enzymes, then adding more enzyme is going to help. If we have a lot of enzymes and only a little bit of substrate, then adding more substrate is going to help. So if we add more substrate, it'll increase the rate of the reaction up to a point. But like I said before, if we have 100 enzymes and only 10 substrates, then the reaction rate is going to be slow. So we can add more substrates. So if we had 100 enzymes and 50 substrates, then the reaction rate is faster. If we have 100 enzymes and 80 substrates, then the reaction rate is faster. If we have 100 enzymes and 100 substrates, then that's like as fast as it could go. At this point, though, if we continue to increase our substrate, so if we have 200 substrates and only 100 enzymes, then it's not going to increase the reaction rate anymore. So if we have more substrate than enzyme, so once the enzymes are saturated, which means every enzyme is bound to a substrate, if we increase the amount of substrate any further above this point, it's not going to increase the reaction rate. At that point, we would need to add more enzymes. So I guess ideally, the best thing would be if you had the same, you were right here, this is the best, your enzymes are saturated, you have the same amount of enzymes as you have substrate, and that's kind of as fast as you can go. So if we add more enzymes, then we can and add more substrates, then we can increase the rate of the reaction. So another thing that we can do is that temperature is going to be important in affecting the reaction rates. And so as we said before, in a lab, we can increase reaction rates by increasing temperature. And so we can do this a little bit in the body, right? If you have like a mild fever, right, your enzymes will work a little bit better. But remember that our enzymes are proteins, and proteins are very, very dependent on their shape, the three-dimensional shape. And this three-dimensional shape is dependent on temperature. So if we have a moderate fever, the enzymes are actually a little bit more efficient. And so at moderate fever, then our reactions will happen a little bit faster. But if we have really high increases in temperature, it's going to actually denature the proteins and denature the enzymes. And so that's going to lose their function. They won't work anymore. All the proteins are going to kind of melt, and it's going to decrease the reaction rate. So for here, most enzymes work pretty much best at normal body temperature, around 37 degrees C. A little bit higher helps a little bit. So if we have a mild fever, that's okay. It makes our enzymes work a little bit more, mild. But once we get any higher than that, you're like at that 106 temperature, that's not good, right? You're going to be starting to sort of denature and like melt those proteins so they don't work as well anymore. pH is also a big effector in how how the enzymes work. And so enzymes are going to function best at their optimal pH. And so normally, right, the pH of the blood, as we talked about in chapter 2, is around 7.35 to 7.45. And so since most of our enzymes are going to be found in the blood and their body tissues, which are around 7.35 to 7.45, most of our human body enzymes work best somewhere between a pH of 6 and 8. And if we change the pH a lot, it's going to disrupt the electrostatic interactions between those, the three-dimensional protein shape, and it can cause it to denature again. And so if the pH gets too acidic, then it can denature the protein. And if the pH gets too basic, it can denature the protein. So really, we want to keep the pH of our blood the same, like right there at 7.4. We don't want to change it too much. Now, some enzymes, though, are special, and they're going to be modified to work in a special environment. So like in our stomach, we have stomach acid that's around a pH of 2. And so we do have some specialized enzymes that work best at a pH of 2. And so they're going to work best in the stomach. But then if they leave the stomach, they won't work as well at other temperatures. So each enzyme has its own optimum pH. So now I do want to end with controlling enzymes. So we said that in order to activate those enzymes, we need cofactors or coenzymes to be present. And so that's one way we can control them. We can, activate them with those cofactors or coenzymes. But we can also inhibit them. And inhibitors are going to basically turn them off. So inhibitors are going to bind to an enzyme and prevent them from doing their job. And so this is important because sometimes we want the reaction to happen fast sometimes, but not all the time. Like once we have enough product, we can kind of turn off that assembly line, right? We don't need to keep making more products. Usually the inhibitor binds to the enzyme for a little while, and later it can release, and that will let the enzyme work again. So it's like when it's bound, we turn the enzyme off, and when it is not bound, we can turn it back on again. Now, there are two types of inhibitors we'll talk about, and I think Bozeman Science, that I linked as an ed puzzle, goes through enzymes pretty well, and it talks about these different types of inhibitors. So inhibitors can either be competitive or non-competitive. So a competitive inhibitor means it competes with the substrate, and by that I mean both of them bind at the same active site. And so competitive inhibitors generally resemble the substrate, which they would have to in order to fit into that active site because there's this lock and key. So this inhibitor is going to be a similar shape to the substrate, and it competes with the substrate to bind at that active site. But when the inhibitor binds at the active site, instead of having a reaction happen, it kind of turns off the enzyme. It's like a cap. It covers it up. It makes it so no other substrate can get into there. And so because this is competitive, if we have a lot more inhibitor, then the inhibitor is going to win out, right? And the enzyme will be inhibited. But if we have a lot more substrate, then sometimes the substrate can beat the inhibitor and the substrate can get there. And so if we have more substrate, a competitive inhibitor is not going to be as useful. So the other type is a non-competitive inhibitor. And a non-competitive inhibitor doesn't resemble the substrate and it doesn't bind to the active site. Instead, it binds to a different site called an allosteric site, another site. And so what it does is it binds to that allosteric site, and a lot of times it causes a change in the shape of the active site. And so that change in the shape of the active site then means the substrate doesn't fit. And so once again, this is kind of like a switch. When the inhibitor binds, it turns off the enzyme. It makes it so the substrate does not fit in this active site anymore. Because of this, because they don't compete for a spot in the active site, then it doesn't matter. The substrate concentration doesn't matter. This allosteric inhibitor here, whenever it's present, it binds to the allosteric site and it totally turns off the enzyme. So it doesn't matter how much substrate is there if the enzyme is turned off. So they are not influenced by the concentration of substrate. So the last thing I want to talk about is how enzymes are arranged together. And so usually when we have some sort of reaction, especially if it's a complex reaction like aerobic cellular respiration, which we'll talk about next time, these enzymes, we might need multiple enzymes. So in order to convert the initial substrate to the final product. Like glycolysis uses like a chain of 10 enzymes in a row. And there's a couple of different ways that this can happen. So a metabolic pathway is a series of enzymes. And this series of enzymes is going to be the product of one enzyme becomes the substrate of the next. So this is like the chemical breakdown of glucose is a metabolic pathway. And so we have the initial substrate here is going to then bind to the first enzyme and turn into this, green one. That's the product from enzyme 1. The product from enzyme 1 is then the substrate for enzyme 2, which is going to turn into, this orange one, which is the product from enzyme 2, which turns into the substrate for enzyme 3, and so on. And that substrate from enzyme 3 is then going to turn into the product from enzyme 3 turns into the substrate for enzyme 4, and so on. And so this proceeds kind of in an orderly manner. And a lot of times they might be embedded in the plasma membrane so that, they are close to each other and they kind of stay close together. Now, we'll come back to that picture in a second. The opposite of this is a multi-enzyme complex, and this is where these enzymes are actually attached to each other. And so they're kind of attached in one big glob, and they work in a sequence of reactions. And like pyruvate dehydrogenase is an example of that. So there are some advantages for the multi-enzyme complex. So if all of them are directly together in one big glob, it's less likely the substance will diffuse away into a different pathway, right? Because they're all together and the substrate will go through very quickly. And then this single complex can be regulated rather than individual enzymes. So we only have to regulate, that one complex instead of regulating, each different enzyme. Pathways, those multi-enzyme or the metabolic pathways, instead of regulating each individual enzyme in this multi-enzyme pathway, so like look here, we don't have to necessarily turn off each of these. Our multi-enzyme pathway is pretty cool because it can actually be regulated by negative feedback. So the first enzyme in the pathway here has an allosteric site. And as we said, they kind of go in a row. So the substrate comes in and it turns into something else and it keeps going and it keeps going. And at the end, enzyme 4 releases the end product. So this is the end product. This is whatever we wanted to make. And then this end product here is going to go back and it's going to then be the allosteric inhibitor for enzyme 1. So once we have enough of the end product, once the end product starts building up, some of it is going to then be able to bind at the allosteric site for enzyme 1 and turn off enzyme 1. So once we have enough product, we don't need to make more product. The product can go back and it can bind to the first enzyme here. And so when that happens, the initial substrate will never be turned into the intermediate substrate. So there won't be any green cells. Like there won't be this green triangle, there won't be this orange square, there won't be this, blue hexagon. And so we don't actually have to turn off these other enzymes necessarily, because if we turn off the first enzyme, then those intermediate substrates are not going to be made. And so it kind of turns off that whole pathway. So another way we can regulate enzymes is by phosphorylation or dephosphorylation. And so phosphorylation is the addition of a phosphate group. And this is performed by protein kinases. And this turns off some enzymes and turns on others. And we'll talk a lot about protein kinases. So adding and removing phosphate groups are pretty important for a lot of things. Dephosphorylation, as the name implies, is the removal of a phosphate group. And it's performed by phosphatases. So kinases phosphorylate things, phosphatases dephosphorylate things. This, once again, just like phosphorylation, turns on some enzymes and turns off others. So drugs can be really useful as enzyme inhibitors. So we can look through and we can figure out how to increase or decrease a specific enzyme activity, and this can sometimes modify homeostasis. And so, for example, penicillin actually targets a bacterial enzyme, which slows the spread of infection. So if the penicillin inhibits the bacterial enzyme, it's going to slow down the reaction in the bacteria, and that slows the spread of infection. Sildenafil, or Viagra, inhibits phosphodiesterase type 5, right? And phosphodiesterase type 5 is a specific type of enzyme, and by inhibiting that enzyme, it's going to cause vasodilation of the blood vessels in the penis. So we can use, a lot of drugs are going to target enzymes and either make them more or less effective. And so, for example, lactose intolerance, right? So we said a lot of times lactose intolerance happens because an individual does not have that lactase enzyme in those microvilli, in the brush border. So lactase is required to break that bond into lactose and form glucose and galactose. A lot of older adults don't have that lactase anymore. It doesn't persist. And so you can treat that with taking Lactaid, which has lactase enzymes. And so if you take the lactase enzymes along with drinking, milk or whatnot, it's going to help break down that lactase. So the last thing I want to mention, and I don't have a picture of it here, but I did put it in your, it should be up. I'll put it up for you. In the book, you have a nice table that talks about all the different types of enzymes, and it gives examples of all the different types of enzymes. So naming enzymes is pretty important. Anytime you see something that has an ASE at the end, ASE, lactase, it's usually an enzyme. And if you look through, your book has a table of all the different classes of enzymes. And I'll see if I can, at some point, you can look through. I don't remember the number of the table. And that enzyme is going to be really important knowing the names of enzymes or the type of enzymes will be really important.